I'd like to welcome you all to this virtual meeting of the Royal Statistical Society's Professional Statisticians Forum. My name is Stephen Pyke, and I shall be the chair of the meeting today. I shall introduce the speaker in a few moments. Just in terms of myself, though, as well as being the chair of this meeting, I'm also the chair of the RSS Professional Affairs Committee. And for my day job, I work at GlaxoSmithKline in R&D, where I'm the head of quantitative sciences. Just by way of preamble, so as some of you may already have discovered, the slides that will be presented today can be downloaded from our website, which is www.rss.org.uk/PSF, the initials of Professional Statisticians Forum, in case my pronunciation is not clear. And those have been available for the last few days, and indeed will remain available after the meeting if you wish to download them later. I should say also, for those who may be following along with paper copies of the slides, that the speaker will call out slide changes as the presentation progresses. So you should find it easy to keep track of where we are in the presentation if you're listening only to the audio portion of this meeting. Some of you will, I'm sure, however, have dialed in, will have joined us via the web portion of the meeting. And if you have, you should be able to see the title slide on your screen now. Those who would like to do so and haven't yet, you still have a few moments to join us on the web portion of the meeting. And again, it's at the same website. If you go to www.rss.org.uk/PSF, you'll find a link to enable you to join the meeting. Just in terms of the structure of the meeting, as you heard with the automated announcement, we're recording the meeting. The intent is to make this a podcast and to post it also to the same page of our website. That will be done shortly after the meeting itself. There will be an opportunity, and it's the nature of this meeting, for it to be, I hope, engaging and participatory meeting. And so if you wish to ask questions, there are two ways you can do so. You will be able to unmute your telephone, and you can do that by typing in *6 on your keypad, and that acts as a toggle. So if you type in *6 again, it will mute your line once more. And just in the interest of keeping background noise to a minimum, we certainly welcome it if, having asked your question, you can mute your line again. So that's *6, which is a toggle. The questions will come at the end of the presentation, I should say. So our speaker will give their slides, and just in the interest of simplicity of managing the technology, keeping the noise to a minimum in the background, all questions, if you can just hold those to the end of the meeting. But there are two ways in which you can participate with questions. As I say, you can ask on the telephone using *6 to mute and unmute your line, or you will find a Q&A button at the top of your screen if you're using the web portion of the meeting. By clicking on that, a box will open up, and there'll be an opportunity to type your question. If you do that, you can do that at any time during the meeting, and we'll pick it up and we'll pull on those at the end of the presentation and call out the questions that have been posted using that facility. I should say that if you post anything, we pick up automatically your name, but it would be very helpful if you, giving a voiceover question using the telephone, if you could also introduce yourself. There's also, I should just mention, finally, a poll button at the top of your screen. For those who've not used this technology before, that's simply an opportunity at the end of the meeting for you to post any feedback. We certainly welcome feedback. We run these meetings on a quarterly basis or thereabouts. And of course, feedback helps us ensure that the presentations are good for the audience, that they're pitched appropriately, but also helps us with any technical difficulties so we can identify those and resolve them for next time. So any feedback would be gratefully received. So all that preamble said, perhaps just move to the meat of the meeting, as it were, and to introduce our speaker. So there will be just one presentation today, but it's co-authored, and the two co-authors of the presentation are Dr. Bretz and Dr. Bjoern Bornkamp. Dr. Bretz and Dr. Bornkamp both work for Novartis, which is a pharmaceutical company, and they both sit within the R&D division of that company. Dr. Bretz is the head of the statistical methodology group at Novartis. In his time at Novartis, he has supported methodological development in a number of different areas of drug development, including drug dose finding, which will be the topic of our talk today, but also in a number of other areas, including multiple comparisons and the use of adaptive designs. Dr. Bretz is an adjunct professor at the Hanover Medical School and also at the Shanghai University of Finance and Economics. Dr. Bornkamp, as I said, he's also at Novartis, and he too is in the statistical methodology group, and he's been working there since 2010. In his role within the group, he's had a particular focus on the development and implementation of statistical methodologies relating to dose-finding trials in phase 2 of drug development, and in his role he also maintains the dose-finding R package, which I think he will be referring to during the course of the presentation today. That said, that's probably enough from me, except to invite Dr. Bornkamp to give the presentation, which, as you can see on the web portion of the meeting, is entitled Implementing a Novel Statistical Methodology in the Pharmaceutical Industry. Bjorn, over to you.
Yeah, thanks, Steve. So, hello everyone. I think just get started. This presentation will be about the MCP methodology. It is an approach for statistical analysis of phase 2 dose-finding studies. MOD stands for multiple comparison procedures and modeling. So it's a kind of a hybrid approach combining testing and nonlinear regression. It was developed in 2003 within the Novartis statistical group to improve dose-finding methods, so that was more or less the starting point. And by now, in 2014, it has been adopted in a number of dose-finding trials currently in Novartis, and it is used in most of the dose-finding trials when it is appropriate at Novartis. It's been taken up by other companies, and just very recently, it received a positive qualification opinion by the European Health Authority, the EMA. The first time the EMA has qualified a statistical method, which is quite a good acknowledgement of the methodology. So by now in 2014 it's more or less an established methodology, and in 2003 it was just an idea of a particular thing to do in dose finding or how to improve dose finding. The presentation is really about how it got from some methodology to a really established methodology. So the presentation is about what happened in these years. As Steve has mentioned, I joined Novartis in 2010. I worked as an external consultant on this topic around since 2005. So I was involved at the beginning primarily in the software development related to this, but I worked together with Frank. That's actually the reason why Frank is also here in this room. I worked together with him on the presentation, and he had more insight into the early stages of how things developed, and if there will be questions, he will be happy to answer them as well. I should mention that there were also, throughout the years, there were a couple of people involved of this, obviously, and way too many to mention, of course, but we should probably mention José Pinheiro, who is one of the co-developers of MCP-Mod, and he's still one of, still working on this as well. He's working for a different company now, but still very active with regards to MCP-Mod. And the other important person at the very beginning was Mike Branson. Some of you may know him. Okay, so on slide 3 you can see the outline of my presentation. I will start giving a little bit of background on pharmaceutical industry in general, with a particular view on how to implement innovation in pharmaceutical industry, and then I will talk about the MCP-Mod and how things happened with MCP. I think the starting point for this was an initiative that was started probably around 2001 or 2002 by the biostatistics management at the time. And yes, one second. There's some technical. Was started by the biostatistics management at the time, and to understand the motivation behind that initiative, I will talk first a little bit about phase 2 dose finding studies in general and the objective in these studies. Understand why there was a need for developing this initiative. And then we'll talk a little bit about MCP-Mod itself, about technical description of MCP-Mod. I will not go too much into detail about this. And I'll talk about how to actually get it implemented in clinical trials, what were the steps taken in terms of internal and external influencing to get this into the trial. The aspect, of course, at the very end, I will talk about the things that we think were actually the aspects in this development. Okay, just as a background on pharmaceutical industry, what maybe distinguishes it most from other areas in the industry areas is that it's a very regulated environment. But as you can see here on this plot, development of a pharmaceutical compound starts with a lot of compounds being synthesized, and then continuously you screen out compounds that are not so interesting or not working as you want them to work until you maybe have a fraction of 10 out of 1,000 entering the clinical trials. Clinical trials mean that these are compounds actually tested in humans. And the further you get in this development, the longer it actually takes to screen out compounds that do not work. So at the beginning you can realize very quickly that the compound doesn't work. Later on it takes a clinical trial to find out that maybe your compound doesn't work. But if your compound went through all of these phases, in particular phase 3 is the one where you have very big trials and very long trials, but if it got through all these phases and you think it's a good drug, you would submit all your data, that means all the data from the clinical trials and the trials that you have conducted to the health authorities. They will review quality, safety, and efficacy of the application. And they are ultimately the one that grant the marketing authorization for the drug. And only from that time point on, the company will be able to make money with that drug. So before, essentially, no companies do not really earn money with it. So the health authorities have a very important role in this whole development process, because they are the ones to decide whether a drug comes to the market. That means when you view it from the viewpoint of innovation, that also means that an innovation is only an innovation if it also convinces regulators to some extent. Because if you do your trials in a specific way, and you think it's an innovative way, but if the regulators think it's not the right way to do it, it's not really an innovation because it will potentially delay your approval of the compound. That means and that applies not only to statistics, it applies in general to drug development. Whatever you regard as an innovation should also be regarded by regulators as an innovation. So it needs to convince not only decision makers in your own company of an innovation, but also the regulators in the process, because they are ultimately the one to grant approval of the drug. I'm now on page 5. And this, of course, this regulated environment creates a certain situation, because when you are in the development of a certain compound and you have certain options, how you could develop it, there might be a more conservative option, how it's always been done, and then there might be a new option which has certain advantages. In the discussions, an important point is always whether this is an approach that will ultimately get accepted by a regulator. If this is questionable, then the typical approach is to use or to rely on established approaches. And that's just how the situation, it's just a product of the situation. Of course, I should say that within pharma industry, there are possibilities to talk to regulators before the ultimate registration. So you can talk to them before that, and you can talk to them on what type of studies you plan to do, what type of methodologies you'd like to use, but that's to some extent also limited. Another challenge for innovation that I don't think it's specific to pharma, but that applies, I think, to statistical innovation, is that, of course, you need to convince the project team members within the clinical trials, and of course, ultimately also more senior decision makers from the benefits of your new approach. And typically, these people not necessarily have a quantitative background. Another thing, of course, is that there is always in big companies a tendency towards standardization of workflows, and innovation changes workflows, and at the beginning initially requires some more work. But I don't think these points are specific to pharma industry. Okay, so let's get into dose finding and what dose finding trials actually are. So you see we're in phase 2, we are not in phase 3, and the role of phase 2 is essentially to select the doses that you would like to test in larger studies, and the type of how do the data look that you obtain in these studies. So typically you will have observed something like this. So you have multiple doses in your study, maybe 3 or 4 or 5 doses observed in the study. You have typically also a placebo, and that's shown on the x-axis, and on the y-axis you can see some kind of response measurement that measures how good your drug performs. The first question that you might have in this situation, well, is there a dose-related effect at all for this treatment? I mean, previous experimentation and previous trials would typically suggest so, otherwise you wouldn't do the trial, but still you would like to be sure before going to phase 3 that this drug is actually working. Then you're interested in, if it works, what kind of effect can you expect, and how does it compare to alternative medications that are maybe already on the market? There's no point in developing it further if it's not comparing good in this respect. And also you're ultimately also just interested in the shape of the dose-response curve. So where does the curve start to plateau, and where's the increasing part of the dose-response curve? And these type of aspects are important in dose selection. Of course, you're not only interested in the efficacy of a compound, but also in safety variables and tolerability, dose response curves, because ultimately a dose selection will always be a trade-off between efficacy and safety and tolerability. So the ultimate aims in phase 2 are really to be able, so if you've done the trial, you would like to know, should we go in phase 3 or not, and you would like to have sufficient data to make this decision reliably. If you decide to go in phase 3, you should be sure which dose you are able to choose for phase 3. That means which dose has the best benefit-risk relationship. So the trial should be informative to be able to make these decisions. On page 7, I'm now on page 7, and the motivation for that dose-finding initiatives that I discussed earlier, that was mandated by the management at the time, is that there was a mismatch between the real objectives of these studies and the actual objectives in the protocol. So the statistical objectives in the protocol were typically ones about testing hypotheses. That means you would test controlled versus active doses. And the study design was typically determined by this objective. That means the sample size, the number of doses that you look at, the dosages used would be chosen so that you can achieve this objective in a good way. If you look at this, what is the output of such an analysis, you can see in the lower bottom, there's a typical output of such an analysis that's actually real data output. So there you can see that was a dose-finding study with 4 active doses and an active control. So AC is an active control, competitor compound. And what is plotted is the difference to placebo. And the main conclusion, if you look at this plot, is, well, all are significant, different from placebo. But there's not much more that you could get from such an analysis. For example, you don't really learn much about what happens between the doses, how is the shape of the dose-response curve. Of course, you can do some eyeballing with this plot, but not really much can be said about this. It's also not clear, for example, which dose of your compound that studies would give an equivalent effect compared to the active control, which is an important question if you, for example, start planning a phase 3 trial, because it would be a lower bound of what you would use typically. On page 8, you can see essentially the same data, but now analyzed with a nonlinear regression technique. So in this case, it was just an Emacs model that was fitted to the data. And you can see that by doing this, you can obtain more information. First of all, what you can see is that you would use your dose estimates. Means you interpolate all the information to be able to come up with an estimated dose-response curve, and this will decrease the variability of your estimates and ultimately also lead to a increase in sample size, of course. You can also interpolate between the dose levels, and you can calculate confidence intervals for quantities that you're interested in. So what you can see here is the red area is the point estimate and confidence interval for the active comparator in the compound, and you can actually also calculate the confidence interval for the dose that achieves an equivalent effect to the active comparator. Now, it's not that these type of methods were not done before MCP-Mod. They were always done, and they are also still always done, but typically only as a supportive analysis. That means the design of these studies was not tailored to these type of analysis. And there are also reasons why this was not done as a primary analysis, because specification of this one dose response model at the trial design might be difficult, because in a clinical trial protocol you would write down before you do the trial what kind of analysis you would use at the end. And specifying this one dose response model is in some situations challenging. On the other hand, if you don't specify what exactly you will do with your modeling, you might end up in a situation where you overfit the data. That means you fit too many models and might end up with conclusions with error. Okay, so ultimately, one of the aims of this informal initiative or biostatistics initiative, dose-finding initiative, was that the statistical objectives closer to the real objectives, and it was identified that dose response modeling was one of the ways on how this can be achieved. And of course, this will have an impact on the study design. You could, for example, use more doses because you share information between doses. You could use less patients per dose. You could investigate a wider dose range, and so on. And initially, primarily were people from the statistical methodology group within Novartis worked on this initiative, but I think almost 1 or 2 years later, this was branched out to the general statistic group and implemented in actual clinical trials. I should mention there was in parallel another group working on improving phase 1 dose-finding designs. I will not talk much about this, but this is also a very successful initiative. I will come back to that later. Okay, on page 10 you can see the first action items. Actually, Frank was able to dig out some of the documents that were used at the beginning to plan the activities, and I just copy-pasted some of the things that were in this document. So at the very beginning, people were trying to try to understand what the current practices are, example trials, talk with people in the therapeutic areas, have contact persons in the therapeutic areas, have a collaboration with the protocol review committee, and so on. And there was also literature review ongoing. So it was not planned, actually, that there will be development of new methods. It was more like what are the methods available and what is the best method that we should be able to, or what we should recommend to use within Novartis. On page 11, you can see a high-level overview or the motivation for developing MCP mods. So as I said, there was a trend towards using more dose-response modeling in phase 2, but according more rigorous standards. And one part of that is that you try to acknowledge the model uncertainty by specifying a candidate set of models at the design stage, and you also specify at the design stage how you will select your models. And then once you have obtained the data, the procedure is built up from two parts. First of all, there's an MCP part, and that essentially is just a trend test that tests whether the dose-response curve is flat or it's not flat. And then the second part is a modeling part, which is really fitting the nonlinear models that you have specified and perform the model-based inference as I've shown you before. So at the beginning, one of the important aspects in presenting that was that's a kind of a hybrid approach that combines two traditional approaches. So in a way, combining the best of two worlds and getting rid of the not so good things. I think actually that was a good idea because at the time, or I think it's still at the moment, there exist two rather separate approaches to dose finding, namely the testing framework and the modeling framework. And I think the MCP mod methodology tries to combine or to bridge both approaches into one methodology. Depending on who you talk to, people value different aspects of the methodology differently. Okay, on page 12, you can see a flowchart of MCP mod just to show you how it would be implemented within the trial. So at the trial design stage, you would select your set of candidate models that would describe what you think could be the potential dose-response relationship for this compound. So that would be based on available information you have from earlier testing. Then you would come up with those potential dose-response shapes, an optimal test procedures based on these dose-response shapes, and ultimately you would perform sample size calculation and determination of the design based on what you think is likely to occur in the trial. When you have conducted the trial, as I said, there will first be the MCP step, assessing whether there's a dose-response signal or not, and then there will be the modeling step where you fit the dose-response models to the observed data. Okay, so this was more or less there already around 2003. I think the paper was published in 2005, but the methodology was available around 2003. The question now is how got it implemented in clinical trials. I think the first people to convince of this, that this is a useful approach, obviously the biostatistics organization. So in this respect, it actually helped that the initiative was mandated by the biostatistics management, because that also meant that the biostatistics management was always aware of that and helpful in this regard. But I think more important aspect was also to get the lead statisticians on board. And by this I mean those statisticians who have review roles within the organization. So that could be, for example, statisticians who supervise trial statisticians who have an oversight of a couple of statisticians. But there are also other statisticians, for example, at least at Novartis we have clinical science review boards who see all the protocols before they get implemented. And they see these protocols at a stage where changes still can be made. So these are quite important. And there are also other review boards existed throughout the years. And I think in many situations it was these lead statisticians that helped in getting MCP-Mod into these projects when they thought it's appropriate. Another aspect is, of course, get a buy-in of the statisticians that actually implement these methods, so the statisticians on the project teams, because they are the ones involved in the discussions with the clinical teams and more senior decision makers, and they have to feel comfortable in what they're doing in the type of methods that they're using. And obviously there's a need for support and technical enabling. I will come to that later. But I should also mention that there was never a mandate for trial statisticians to use particular methods. So it was never mandatory, for example, to use MCP-Mod. It's always at the very end up to the statistician to decide whether it's appropriate or not. And there have been situations where we also said, well, in your trial, maybe it's not the right thing to use MCP-Mod, maybe to use something different, of course. But yeah. Okay, of course, company internal not only means the statistics organization, but also, a whole lot of other groups are involved in this. There were a lot of cross-functional initiatives to improve dose finding, and the biostatistics organization was always part of it. So in 2004, there was one initiative that was quite impactful because there were decision makers from all different areas involved, also from biostatistics, but also the clinicians, safeties, pharmacological modeling, and they developed ideas to modernize drug development. And of course, this biostatistics person would think that within dose finding, for example, MCP-Mod would be a good approach to improve the finding studies. And a similar initiative later on was the Get the Dose Right initiative. And another example is in 2011 there was a training created for clinical decision makers on how dose finding should be done. And there were obviously by the MCP-Mod team various presentations to non-statistical groups throughout the years. On page 15, in terms of technical enabling, of course, as I said, statisticians need to know how to use the methodology, and they need to feel comfortable in using this methodology. So there were training sessions in 2005 and 2006, and later on repeated 2011, for example, on the methodology itself. But throughout the years, there was also and is also hands-on support for statisticians. That's, for example, one of my main tasks at the moment. So when there is a dose-finding study, I would get involved and work with the statistician together in all the aspects that are important in planning this trial. For example, in discussions with the clinical team, help in what to write in the protocol in the statistical section, how to write the analysis plan, and how to implement the methodology in computer code. And that's another important aspect. So software, there was since 2005 there was a validated S+ library. Now we have SAS macros available that need to be fine-tuned for each situation. And since 2010 there's also the dose finding R package available that has MCP-Mod implemented, but also other things. And of course, in terms of this technical enabling, also another aspect is extending the scope of the MCP-Mod methodology. So originally in 2003 it was developed for normally distributed data, and through the years we've extended it in various directions just to be able to use it also in other situations, so binary data, time to event, and so on. But I think in pharma industry, I'm on page 16 now. In pharma industry, what is also important is this external focus, and I think the group always considered it is also important to get feedback from the scientific community. That means, to some extent, publishing the methodology also in papers, because this provides an independent and external scientific validation of your own ideas, and I think this is quite important because it also generates good arguments for internal influencing. For example, if there is a trial statistician and he or she is unsure whether to use the method, if you say, well, we have published it in a very good journal and the reviewer liked it, it's, I think, a good argument for them to use the methodology. And I think it also helps from a regulatory perspective if you have a methodology implemented in a clinical trial and you have a reference to a good statistics journal in there, it might convince also the regulatory statistician. I don't think it helps too much in convincing clinicians or the clinical team of the methodology. But what it helped more in this regard is, I think, also that there were big across-industry initiatives. The pharma group was a group of statisticians from different pharma companies that worked on how to improve dose finding. And what they did essentially was a very big simulation study comparing different new methods for dose finding. And what was powerful about this initiative is that there were white papers being published, 2007 and 2010, and these were discussed by regulatory statisticians with rather positive feedback. And at the time, I think it was, for example, the position of many of the health authorities was not, for many people, maybe not 100% clear, for example, whether you can actually use these model-based approaches like MCP-Mod or if you can use Bayesian approaches in these phase 2 type of trials. And given the feedback in this discussion, I think it became clear that actually it's possible to use these methods. And I think this helps in particular, for example, in the clinical teams, a regulatory person in the clinical teams would get convinced by showing these discussions of these regulatory statisticians. I think another aspect was also attending external conferences and giving external short courses at these conferences. So I estimated that there were around 6 to 8 conferences, short courses on MCP-Mod throughout the years at different conferences. And typically these were attended, of course, by statisticians from academia who might have good feedback on the methodology, by industry. And for example, there might be people, and there are actually people who attended short courses and later implemented the methodology in their trials. But it's also interesting because there might be regulatory statisticians attending these short courses and have feedback on the methodology. But actually one of those short courses where the idea of this qualification of what came from. But these external arguments, although they at first sight they are maybe not so powerful to use in a company internally, but I think ultimately they turn out to be quite powerful. Of course, there were also other important factors that helped us, although we were not really so much involved in that. For example, a good way of convincing teams in discussions, in team discussions, is that there were rather prominent examples within Novartis where those findings simply went wrong, and it was done in a way that wouldn't have happened if for example, MCP-Mod would have been used. And these were rather prominent examples, so people would know about it if one would ask about it. And partially it was also because the M&S group, so the pharmacological modeling group, did a training in around 2010 that was called Get the Dose Right. And every Novartis pharma associate would attend this training and would know about the topic. And I think that created a good starting point for a discussion in clinical teams, because you could assume that most of the people on the team have attended the training and would know about the issues and the potential improvements. Okay, so on page 18, I'm coming to the end. Some remarks. I mean, I have to say, for Frank and myself, it was a very interesting experience to compile these slides. And looking at it now, be probably a little bit more planned and smooth than it actually was. Because one knows who works in pharma industry or in a big company knows organizational structures change. But I think the important thing here really was, and it was only at the beginning a real formal initiative. Later on it was more like that there was always a team of people, a core team of people that felt responsible for the topic and also was regarded as being responsible for the topic of those findings. So that would, if someone would have a problem with a phase 2 trial, they would know whom to contact. And as I said, it's more than just Frank and myself. There were always more people involved. There was also there's also a dose finding focus team working in the background with roughly 5 to 6 people who were involved in this. And yeah, people changed throughout. In hindsight, I think most important, or the core reason or the key aspect, was probably that there was a core team of people who was able to spend time on this continuously. Because I think in pharma industry, things usually don't change very quickly. Change takes some time. So you also need a team of people to be able to spend some time on this to be able to make a change. And I have to say, we were in the lucky situation that we were always supported by the biostatistics management. I mean, in different forms, but there was always a support from the biostatistics management. Another aspect is that I think not only important, to have this internal focus, but also this external focus, because this external focus can result in quite powerful arguments also in internal influencing. And I think the EMA qualification opinion that we received beginning of this year was very powerful in this regard. And coming back to the other initiative, the phase 1 initiative, essentially it also turned out that one of the key aspects for them was also having this core team being able to spend time on this continuously. So this was also quite interesting for us to see that although the team somehow split a little bit during the times, there were several conclusions in how to get these innovative methods implemented. And I think that ends the presentation part, Steve.
Well, thank you, Bjorn. Very clear and very enjoyable presentation. I'm sure there'll be some questions. Just before we come to those, just a couple of reminders. Well, firstly, actually, I perhaps should have said earlier on in introducing Bjorn, of course, what I forgot to mention was that he was, on the basis of this work, the winner of the 2013 Prize for Statistical Excellence in the Pharmaceutical Industry, and that's an annual award sponsored by jointly Statisticians in the Pharmaceutical Industry and the Royal Statistical Society. So this talk arises from some of those the experiences which were recognised in that prize. Just to remind you then, if you have any questions, you can open your line by punching in *6 to your keypad. If you do wish to ask a question that way, please introduce yourself, and when you've finished, please *6 again to toggle your line to mute to keep the background noise down. You can also use the Q&A button on the web page if you're joining us using the web features of this meeting. Maybe just to get the conversation going, Bjorn, maybe I'll begin with one. As you know, I work in this area a little myself, and one of the things that struck me was that if there's any challenge in getting the support of the organization to this shift, it really comes to the fore when you move away from conversations around what way to analyze data and start talking about the way in which a different analysis is actually informed by a different design. Some of the things that this might give rise to as a methodology would be thinking around a different way of allocating patients across doses, a different number of doses, a different size of study, and so on. I wonder if that was your experience also. Was this really if you like, the rubber hits the road when you start changing the design of the study? Or were there just as many questions and conversations around the analysis?
Yeah, I think that's a good point. I know that there are examples, for example, if you have a design more or less fixed, and you just use MCP mod instead of a pairwise comparison, then in some situations the clinicians might not even notice that there is much of a big difference. But it's really in the design stage that this type of analysis methodology can make a difference. For example, a traditional kind of thinking in clinical teams is that, well, if we add one more dose, we have to add, the same amount of patients if we add this dose. And so we don't want to have another dose. But with a modeling type of approach, you can say, well, you can have one more dose, but we just have fewer number of patients within the dose arms. So I think that's really where it makes a difference within the design. I think that's a good point, yes. Yeah.
Very good. Well, I'd like to welcome others who may wish to ask a question. I see a question's been posted. Let me, just while others do feel free to come in with a verbal question on the line, but here's one that was posted a few moments ago by Jean-Christophe Lemarie. And he says, on slide 2, you say that the methodology is used in most of the Novartis dose-finding studies when appropriate. I wonder if you can give some examples of situations where MCP mod might not be appropriate.
Yeah, I mean, I haven't talked really much about methodology itself, so it's hard to. The answer is no more for the people who really know MCP mod. And one limitation, of course, is that MCP mod focuses around dose response. That means you need to have some kind of dose variable somewhere in your model. For example, if you have 3 or 4 treatment regimen, or if you have 3 treatment regimen and end dose, then I would find it difficult to recommend using MCP mod. But we have actually, if I don't know if I can, there is this in the EMA qualification opinion. There's a section on the scope of MCP mod, and I think I don't know if I can post the link somehow. But that's
that might
be well.
Why don't we aim to do that, Bjorn? Perhaps after the meeting you could share something with myself and the meeting organisers, and we'll arrange for something to go up alongside the presentation. Very good. Anyone like to give voice to a question over the telephone line?
Yes, I have one question from Pascal Gerard-Mercerono.
Go ahead.
Okay. Well, first of all, thank you for a stimulating presentation. So my questions concerns, do you have an experience with joint modeling of adverse effects and efficacy? And well, in this case, the dimensionality of the MCP increases due to the multiple dose response and efficacy those adverse events response. How do you deal with this multidimensionality?
We have, at least in the type of MCP mod that you specify in the protocol, we haven't really pre-specified these type of analyses. So trading off efficacy and safety is something that we have not yet done as a pre-specified methodology. Of course, we did some modeling, trading off or jointly doing adverse effects and efficacy, but that was never done as a pre-specified analysis. So typically in the protocol, one would focus primarily on the safety and plan the study based on the efficacy, sorry.
The efficacy, yeah.
And you would plan the study primarily for the efficacy, and then do this type of modeling at the end after the trial, but not specify it formally, at least.
Okay, thank you.
Yeah, I mean, this sort of gets into an interesting area. It seems to be perhaps going slightly off topic, but it's the extent to which one sees these kinds of studies as learning or confirming, to use the jargon from the pharmaceutical industry, and to what extent is this an opportunity to learn And if it is, how much freedom and flexibility should one allow oneself after the fact, as compared to phase 3, where we're very much in a confirmatory space. So, I think in a sense the expectation that you will be able to pre-specify absolutely everything at such an early stage development is quite ambitious. I had another question to you, Bjoern, which was about the sort of, I suppose it's about the journey that you've been on, sort of going back to the sort of nature, the process by which you embed and implement these types of approaches. You said early on that in these studies, which I guess we might describe as being primarily, the primary objective might be to characterize dose response, and yet the analysis didn't always directly reflect that. What you might have wanted to do, perhaps this was the first step, would be to say, well, a great first step would be to ensure that the primary analysis reflected the primary objective. That in a dose response study, which was aimed to characterize the dose response, we did that, and that would lead you to using models. Now you've gone another step further, which is to say, and moreover, here's a class of model or an approach to modelling which we recommend. But I wonder, going to your sort of closing remarks, how much of that sort of two or multi-step approach becomes apparent at the end of the journey, or was it actually a more of a one step at a time approach, and the first step was to just get folks thinking much more about using these models of any description, of any class, of any type, in the first instance as the first step on that journey?
I mean, I have to admit this is my interpretation. Maybe Frank has more to add to it, but my interpretation was that at the time it was considered too risky to use dose response modeling, just specifying one dose response model. And this is the whole reason was that this was the whole motivation for developing MCP-Mod. So there was, you're right, it was exactly like that you would try to get the objectives, the statistical objectives of the trial closer to the real objectives. That means using more of dose response modeling, but at that time it seems that people conceived that it's too risky to use one dose response model, and that's why the reason why MCP-Mod was developed and then used.
Any other questions from anyone? I see we have one posted. I don't know if Roger Rowan is on the line and would like to give voice to his question. Are you there, Roger? If not, well, maybe I'll just I'll read it out for you, the audience. And he says, although the Emacs model looks adequately flexible, how can you be sure that it is in some way influencing the solution? How do you know that a variant of Emacs won't give a different answer? So I suppose this is sort of building off what you've just said to some extent, Bjoern. Maybe you'd give some thoughts on Emacs and its usefulness or the lack of usefulness in some settings.
I think I should maybe have presented more on MCP-Mod, because within MCP-Mod you are not confined. You don't need to use only the Emacs model. You can use multiple models. That's the whole idea of MCP-Mod, that you can say, well, in many situations the Emacs model is actually the one that's right to use. You can safeguard against this specification by using multiple different Emacs models, dose response models. You can see it maybe on, if I go back, in this example, so you can see that you might, for example, also specify a linear model or a model where the downturn of the effect at higher doses, or other kind of models. Essentially, everything that you think is plausible at the design stage, you can specify as candidate shapes. So it's not confined to the Emacs model.
Any other questions from anyone?
Hi, Steve. Can you hear me?
Yes.
Hi, it's Nigel Dallow here from GSK. Thanks for a lovely talk, and this is a tool I've used a few times now, so I'm starting to get some familiarity. But my question would be around how this method, using MCP MCP mod would compare for a user-defined approach where say you use, say, DIC or other statistics to compare, I don't know, 2 or 3 models? What is there anything additional that MCP mod will deliver you, will help you, than I guess doing some of that outside of the system?
I think what you mean formally, what the additional step is multiple comparisons. I have to say that how important that step is different between projects. In some situations, for example, you know before the trial starts that the placebo response will be very low, and the MCP step is not important at all, and it was not really important. And then the MCP mod procedure more or less reduces to what you have just mentioned, like fitting 2 or 3 models and then selecting one, or performing model averaging based on these models that you have. In other situations, the MCP step can be also quite helpful, particularly if the compound really doesn't work well. Because the MCP step is something very, very basic. It's just looking at whether there's a dose-response signal or there's no dose-response signal. And having, in some situation, having a p-value for this comparison is helpful, in particular if the dose-response curve is flat.
Okay, thank you. Nice answer, thanks.
Hello, this is Anja Victor. I got a question. You mentioned that you had 2 examples where the dose modeling went wrong in the traditional way. How did you find out?
These are complicated. These are complicated examples. Yeah, I could have 2 talks on both of them, but it was not that the dose response modeling went wrong. It's much more complicated. It was the way the dose finding studies were conducted, which created problem in phase 3 with accepting that the regulators did not, well, in one case they did accept the dose that was proposed, but in the other case they did not accept the dose that was proposed for the phase 3 trial. And that created problems, but it's too complicated to explain now via the phone. But essentially you would have to the principles underlying MCP mod, which is somehow performing a dose-finding studies that gives you information on the dose-response curve. You wouldn't have had these problems that they faced in these 2 examples. Essentially, in one of the cases, the problem was that they didn't do dose-response modeling. It was a very, very small phase 2 study, and that created the problems. Very few doses that created.
Bjoern, just to build on that a little and take it in a slightly different direction, I was wondering when I saw that reference in the slide deck, whether you think there is more power in the sort of negativity of a failed study or a negative outcome or something which in some sense the organization regards as being a disappointment. Is that more powerful in galvanizing change, or is it the power of persuasion, the gradual process of engaging and teaching and so on? Or do you need both? I mean, I'm just curious, how important were those failures, as you refer to them, in gaining acceptance for the methodology?
I don't think they were important in gaining acceptance for the methodology. It was more like that people would know it, and that is an easy way of getting people into the conversation about these topics within the teams, because particularly if you're talking to non-statisticians, you need to find something to get the conversation started. And this was an easy topic because they would know about it.
Mm-hmm. Anybody else with any questions? We have a few minutes still. I'm happy to take some more calls on the line, or if you want to post anything. I can keep asking questions all afternoon, I'm sure. I've got another one, but
Steve,
if you have any questions, jump in.
Steve, I've got another one. I was on mute. I was talking to myself. So I just wanted to get an idea from yourself around the selection of the models that you could potentially use for this approach. And it strikes me that from experiences I've seen within GSK anyway, there's sometimes a risk, unless you select the models very carefully, that unusual data can drive the final model that you go with. So do you have any recommendations that you could help us with in terms of how upfront you select the models that you plan to put in for this? And the context beyond the example I'm familiar with is where I've seen, biologically I don't believe the response is going to fall off at the end of the dose response, but the last dose just by chance gave a lower value, and the person putting in the different functions have actually chosen an umbrella function as well, I think as a beta function. So it feels like you need to be careful, but you also need just to think very carefully through how you select the models and which ones to select. So do you have any guide or any context that you could help us determine what models to put into this as we go forward for different molecules?
Yeah, that's actually the example that you just have said is actually the perfect example for this situation, where you have just chance the highest dose is a little bit lower than the next to highest dose, than an umbrella model selected. But I think if you know beforehand that that you wouldn't choose a unimodal model, then you shouldn't include it in the candidate set models. And I think, to be honest, I think the major discussion typically focuses around this, whether it will be a monotonic or it will not be a monotonic dose-response relationship. And having a consensus about this at the design stage might actually help in the discussion after the trial.
So really so not just saying, well, these are the 6 models I always use, I'll put those in, but thinking very carefully around the biology of the molecule.
Yes.
Okay, good. Thank you.
I mean, if I may, this seems to me to get to a very deep and important philosophical issue, if I can put it that way, which is, does one believe the data or the model? Does one put more weight on one or the other? And I think as statisticians, we're trained, certainly those of us who are trained as frequentists, we're trained to believe the data. And perhaps those more the Bayesian tilts might put more emphasis on the model. And I wonder, where you are on that spectrum, Bjoern.
To be honest, I think the MCP mod has both type of aspects, and I think maybe that's part of the reason why it got some acceptance within the statistics community. Because if you're a Bayesian, you might interpret it as a prior distribution on different models. But if you're more a frequentist, you might look more at the MCP part and the p-values that you get. So it's somehow a compromise between.
Very, very political answer.
Bjoern, can I just follow up on my question? You also talk around model averaging, which in the examples you provided, you didn't go any further. That seems quite a useful technique as well. Do you have any feel of whether this has been taken up? I can't remember if that's part of the EMA validation as well. So could you just talk a little bit about the model averaging?
So I have to say I like model averaging. And I like it because you might end up in situations where one model is just a little bit better in terms of, for example, if you look at AIC or BIC, and just by chance one of the models is just slightly better than the other. I think in these situations it's not natural to just pick one model and forget about the other one. So at the moment, at least, I'm tending towards more using model averaging. But yeah, I think within the qualification and within MCP mod, we didn't really say one of the things is better, but the way how someone comes to me now and would like to use MCP mod, I would suggest to use model averaging.
That's great, thank you.
Oh, hi, it's Roger Owen here. I have a question with regard to the candidate models. Although you could have a class of candidate models which looks very rich in the sense that it has all sorts of different shapes on the as you have in your slide, which I've now seen. There would nevertheless be assumptions of the functional forms. You may assume something quadratic or whatever. How do you know that, though? How would you be sure? Is there a way of checking whether, that the particular choice of functional forms isn't in some way restricted? It's a similar question to the one I had before, but slightly expanded.
I think that's part of the model somehow to specify the candidate models that you think are plausible. Of course, that makes an assumption.
I mean, they're plausible in a broad brush sense, but I've seen situations where the exact choice of model, you can have two models which look the same to the eye, they look very similar, but they can give quite different answers.
Yeah, I mean, one thing is, if for example, have a rather broad set of candidate models, then one way of performing inference, then if they are both fitting the data equally well, one way of proceeding then would be to use model averaging, because then you can see that both are valid. Yeah.
Mm-hmm.
I mean, what I have to say with respect to the candidate models and the variability in these trials is typically very big. So that means at the end of the phase 2, the dominating factor will typically not be the bias in estimates, but it will be the variability in estimates. And typically, it makes sense to maybe have a small bias. Maybe your Emacs model is not exactly like an Emacs model, but you can reduce your variability quite a bit and maybe have a bit of a bias in there.
Right. Okay.
Because just variability in these trials is extraordinary sometimes. It's really, really, really large, and it just makes sense to smooth your estimates.
Mm-hmm. Okay, thank you.
Yeah, I must say I'd be with you on that one, Bjoern. I thought you gave a good answer, and I think I would head in the same direction. We've got a question posted, and I think I'm going to be closing the meeting fairly soon, so if you've got anything you'd like to ask and haven't had a chance to do so yet, now's your last chance perhaps. There's a question posted by Mingzhu Hu. Can you explain what you actually do with optimizing statistical tests? Is it via weight selection? Yeah, what we actually doing on a high level, of course I cannot explain it in detail now. Maybe if we send out later on the link to the EMA qualification opinion, there are references to paper on this is actually how this is actually being done. But a simple explanation is that you perform a trend test, and that trend test is optimized for your candidate shapes. That means, for example, if you specify an Emacs model with a certain shape, then the test will be not optimal, but very close to optimal to detect this particular shape. But it is done via contrast tests. I think, yeah, more detail is difficult to say just here. So let me ask again, any final questions before I move to begin wrapping things up? Okay, we're not hearing any then. I shall just begin to draw things to a close. Well, firstly, thank you, Bjoern, and thank you also, Frank, for your presentation today. I think both of us perhaps will have been surprised by the technical nature of some of the questions. This is an open line, and obviously people won't all necessarily have a great deal of experience immersed in dose response studies, but clearly we have an audience who's well versed in those issues, at least some. And we thank them for their questions today. So thank you to our speakers and thank you to our audience also. We do hope you found the session useful today, and if you did, I'll just take the opportunity to remind all of you that this is one of a series of meetings. We meet approximately once a quarter. In fact, our next meeting will be on Wednesday the 21st of May on a very different topic, which is improving the accessibility of official statistics. And then again in June, on Monday the 2nd of June, we'll have a meeting on making a success of a statistical consultancy. So, as you'll gather, very varied topics covered in this series. And you can get more details, of course, from our website, which for the last time is www.rss.org.uk/PSF. As I said, the podcast recording of this meeting will be available on our web pages very shortly, including the conversation and the answers that Bjoern gave and Frank gave to the questions. I'll finally mention that RSS will be holding its conference this year, and we would encourage you again to look at our website for details. It will be in Sheffield City Hall on September the 1st to 4th. And with that, I think I'd like to close the meeting to thank our speakers, Frank Bretz and in particular Bjoern Bornkamp, for their very interesting talk, and wish you all a very good day.